Multicellular organisms are composed of a diversity of cell types, specified in an ordered and genetically supervised way. Acquisition of cell diversity is critical for building functional organs, and when affected could lead to organ dysfunction and disease.
Muscle cells differ by their size, shape, attachment to tendon cells, and innervation. We use a wide range of techniques, including cell type specific, single-cell genomics, genome editing, or confocal microscopy, to understand how these distinct muscle cell properties are defined.
Fine-tuning of gene expression is crucial in defining discrete properties of individual muscles. In addition to transcriptional regulation, we are studying an emerging role of epitranscriptomic regulation.
During development, each muscle needs to find its corresponding tendon cells to attach to the skeleton. This implies that muscles and tendons have to communicate with each other to develop in a coordinated way. In the lab, we use the developing Drosophila leg as a model to identify the molecule that ensure the crosstalk between muscles and tendons during the development.
Muscle size depends on the number of myoblasts that fuse to build it. When myoblast fusion becomes excessive, the multinucleated muscle fibers split. We believe that dissecting muscle fiber splitting in Drosophila may help to understand and find a cure for pathological splitting observed in human dystrophic muscle.
Drosophila cardiac tube represents simply but fully functional organ with the heart beating parameters regulated by evolutionarily conserved genes. Identifying gene deregulation that underlie cardiac dysfunction in Drosophila is thus the way to better understand molecular determinants of human heart disease.
We applied in vivo imaging to track cardiac cells during Drosophila heart formation and to make links between gene expression signatures, the diversity of cardiac cells, and their collective migration. We use also the power of Drosophila genetics and Drosophila models to understand gene deregulations associated in cardiac defect associated to myotonic dystrophy type 1.
The goal of our researches is confirmation the variant mutation with unknown significance in human.
All in all, we investigate mechanism of development of heart and muscles, and Drosophila is our favorite model organism in which assess
How the diversity of muscle and heart cells is acquired.
How the development of muscle and tendon cell is coordinated to generate a functional muscle.
And finally, how the pathogenic muscle states arise.